This is a production of Cornell University.
So yeah, I was going to get my picture scanned. I have a picture from when I was an infant standing in front of grad student housing here with my parents. And my PhD advisor was in the picture wearing very nice 70s pants, but I forgot to scan it. So all right, so I'm going to talk today about maize and spaces of the genome. So it's nice to start with pictures of plants and remind you that I actually do care about plants. I won't have a lot of them in my slides later on. This is just illustrating that this is probably the last institution I have to try to convince you that maize has diverse phenotypes and that there's lots of phenotypic and genetic diversity within the species. As a geneticist, what I care about is what are the things or the information that underlies this heritable variation. So that variation is probably hiding somewhere in the maize genome. And we've had a genome in maize for a number of years now. If we look at the most recent version of the genome, so this is true of many genomes, the part that's genic is pretty small, okay? And that genic portion is probably an overrepresentation because I'm including the introns and some of the promoter. The coding portion of the maize genome is a small portion of the genome. The vast majority of the maize genome is intergenic space, either classified as transposon or other. And those transposons are largely long terminal repeat transposons, terminal invariant repeats, and helitrons. So most of you probably know about transposons, but just a quick reminder. In general, they can be put into two classes. So you have class 1 retrotransposons. These usually use an RNA intermediate for transposition and are often copying and adding copies rather than excising from the genome. DNA transposons, the major class we think about are often the TIR elements, but there are also helitrons and other types. These use a DNA intermediate for their movement. In maize, we'll mostly talk about LTR elements and TIR. There are lines and signs. There are a number of helitrons. Both of those types are harder to find, and so it's not that they aren't important, it's just that they're harder for us to work with, so I'm not going to tell you as much about them, okay? Okay, so these transposons that are a big part of the maize genome, where are they? How is the genome organized? So this is a figure from Jeff Benetson's group from quite a number of years ago. And what it's doing is it's taking the whole maize genome and dividing it into 1 megabase pieces and showing you how much of that 1 megabase is transposon. Now, we often think of the pericentromeric region, so near the middle of the chromosomes, as having higher transposon content. You can see that they do. But actually, the thing I care about more in this slide is often telling you that if you look at the scale, it only goes down to about 50%. So even these, quote, gene-rich arms are still half retrotransposon in the maize genome, okay? If we look at this in a little more detail and look at how these genes and transposons are arranged, I'm going to use 2 figures here. So one of these, this one is from a paper from Hugo Dünner, and this one's from a paper from Anthony Rafalski's group. And both of them are showing what happens if you sequence a couple hundred kb of a particular region from multiple haplotypes. So on this top one, all of these things in kind of golden colors here, these are genes, okay? All the colorful triangles are transposons. Down here, it's similar, everything in black is a gene, everything in other colors is a transposon. So there's 2 things I want to point out. Number 1, if I pick a particular gene, so this gene here, TAX7077, it has different transposons inserted upstream of it in different haplotypes. And this is relatively common. I can find lots of other examples in both of them where the type of transposons inserted near a gene is different between different varieties, okay? So not only are these genes flanked by transposons, but they're often differing between different haplotypes. The other thing I need to mention now, because people, we talk about maize, we talk about its diversity, and people often ask me at the end of my talk, how can you even study this? Because things are just moving all the time. So these things historically moved. There were bursts of transposition. As near as we can tell, there's very low to no movement, ongoing movement of most of these retrotransposons in current varieties of maize. So even though there's evidence of extreme instability in the past, things seem to be quite stable now, okay? All right, so told you I'm interested in what's going on in the genome. I have two different snapshots here. And in both cases, the things on the bottom, the blue things are genes. And these are two classic examples in maize of distal regulatory regions. So VGT1 influences the expression of this gene to influence flowering time. This is the TB1 locus that was important for domestication. There's a distal regulatory region. One of the things that I'm really interested in, so I personally come from a perspective that changes in gene expression patterns and levels is critical for phenotypic diversity. And yet, if we're looking for that intergenic space, which is really heavy with transposons and mostly put into heterochromatin in maize, how do we find these regulatory regions? How can we use chromatin and other features to sort through and find those regions? The ability for me to put those yellow boxes on here took people many, many years of careful mapping and cloning of those regions, okay? Okay, so things I want to get at today while I'm talking with you. And I was going to mention this earlier. If you guys want to interrupt me at any time, feel free. I don't need to get to the end. If we don't get all the way through that, that's fine. I'll just stop and release you whenever we're done, okay? So the first question is, what are the sources of heritable variation? So what information is there? What is the role for epigenetics and chromatin beyond the genetic variation we think about? And second, what is it that would drive variation for gene expression patterns? In particular, what are the sources of novelty? If we say that genes are important for, or expression levels of genes are important for controlling traits or responses to the environment, how is it that you would gain new responses to an environmental cue? I will spend lots of time in this first one, and I hopefully will get to the second one. So the first thing we're going to talk about, we're going to spend a fair bit of time on DNA methylation, where it is in the genome, how it varies, and how that might affect function. And then we'll talk about transposons and gene expression. All right, so we'll get into the methylome. And I'm going to give you a little bit more introduction, particularly about methylation and why I'm studying it and try to convince you it's worth thinking about. We'll see if I succeed or not. So methylation, we often lump it together with this word epigenetics. And people use this word epigenetics different ways. I was telling Eric last night, it's tough to try to stand in front of Eric and try to introduce epigenetics to you because he probably does about the best job of that thought pattern. So when I use this word epigenetics, I'm talking about heritable information that is not due to DNA sequence changes. And we can think about this in various ways. We can think about this mitotically. Is there a change in something that can be remembered throughout the lifespan of an organism? You can think about this through across generations in meiotic memory. Chromatin modifications, things like DNA methylation and histone modifications. These are words that often in our mind, we almost lump them together as those are epigenetics. I would suggest some caution in that linking. So in many cases, they are a mechanism of epigenetic inheritance. So if you have a true example of epigenetic memory, often these types of things are involved. Not always, but often. However, these marks themselves are biochemical marks. They can be influenced by the genotype, so genetic sequence can influence them. They can be influenced by transient properties. So expression level of the environment could influence them and cause a change in these things that is not heritable in any way. Okay? So depending on your definition, these marks may or may not fit as being truly epigenetic. Now, I've asked myself a question several times is why is it that we are so excited about epigenetics? Another way of asking this question is why is it that most people are convinced if they can get that word in their title, they'll get into a better journal? Okay. So there are a couple of different reasons I think people are excited about or think about epigenetics as being particularly novel. So one of them I think comes back to how we teach gene expression. So I teach undergraduate genetics, and like pretty much all undergraduate genetics and high school genetics, we teach people gene expression by teaching them the Lac operon. And in the Lac operon, you basically have a simple mathematical equation. If you can predict the presence of a functional cis-acting sequence, an operator, and you have or don't have the proper transacting factors, you can predict whether expression will happen or not. I think that paradigm has served us well. It fits for a lot of things, but when people got to eukaryotes and realized that they could have the right transcription factor, they could have the right cis sequences, and they still didn't get expression, they were fascinated. Why is it that our math doesn't add up? Okay, and chromatin is part of that answer. So chromatin is critical for regulating gene expression, and it plays an additional role beyond the normal cis and trans factors. That view of epigenetics doesn't require any heritability, just that there is additional factors in regulating gene expression. A second reason is this idea of that there is additional information in the epigenome. There's information beyond what we could capture from SNPs. This is often included in this concept of missing heritability, but it could be part of our missing predictive ability would be another way to think about it. The last one is this idea that we could have transmission of acquired characteristics. So this comes back to the whole Lamarckian idea, right? And people are interested in this idea that the epigenome could be influenced by the environment or have different rules of inheritance. People are enthused about this. Often I think that in a lot of cases people have been looking for a mechanism without having a phenotype to explain in that. So I think we'll as we talk, we'll go into a little bit of studies we've done looking for evidence of this. I haven't found a lot myself, but that's not to say it doesn't happen. I think most of what I'll talk about links more to this second part. So in agriculture, if we think about why we care about epigenetics, I would say that in one sense, historically, we didn't need to care. So when selection in plant breeding was done based on a phenotype, it didn't matter what the underlying cause was. We selected for traits. We would capture those traits with varying efficiency. I think that the bigger issue now is as we're moving towards genomic selection and we build models that are partly based on phenotypic data, but also are based on the assumption that a model trained once can apply to other individuals. There's questions of would there be additional predictive information from an epigenome that you would not be capturing with SNPs? So do we get additional information? The other alternative, and the other reason we can suggest that studying a methylome or an epigenome is important, is the idea that this type of data would help us find functionally relevant portions of a genome. So when we have a big species like maize, and I gave you those examples of distal regulatory regions, can this kind of information help us to find those? Okay, so we're going to get into, finally, I'll show you DNA methylation actual data. So I'm going to remind you that when we talk about DNA methylation, we're talking about 3 different contexts. So a C followed by a G, an H, where it's any base other than a G, and then a G, or 2 H's. These sites have different molecular machinery for creating and maintaining that methylation. In each case, there's essentially a self-reinforcing loop. So one of the properties of methylation that makes it stable is the ability for cells to remember it after DNA replication. So this ability to remember comes from these self-reinforcing loops. Sometimes it involves symmetry across a DNA strand. Sometimes it involves self-reinforcing loop with a histone modification. Other times it is a more complex pathway. Methylation we often think about as being associated with gene silencing, and it certainly can be. Heavily methylated gene promoters or transcription sites often are associated with silencing or lower expression of a gene. However, its location matters. So methylation in the middle of gene bodies seems to be tolerated just fine and not influence expression level very much. So to study and relate those things, we need to have a profile of a methylome. And we're doing this with whole genome bisulfite data. We used to do this with microarrays, so some of the slides I'll show you have older data that is less, it does not have context-specific information. There's a whole group of people that have done a lot of work. So this was a collaboration with Matt Vaughn at the Texas Advanced Computing Center. Steve Eichten has done a lot of the things you'll see, as well as Ching Li. Now, one thing I want to bring up is we know a lot about DNA methylation and how the methylome is influenced and what its function is based on Arabidopsis data. Arabidopsis is a relatively simple genome. I want to highlight here and in the next slide why that's useful but doesn't give us quite enough information. So here I'm showing you about a megabase of the Arabidopsis genome or the maize genome. And I'm just showing you two types of methylation, CHG and CHH, as well as a histone modification, H3K9. So first of all, you'll see that Arabidopsis has much higher gene density. There are TEs, but in maize, the TE density is much higher. If you look at these two marks, they're very much co-localized in the Arabidopsis genome. In maize, we see a very different pattern. Virtually the whole genome has this CHG methylation. There are only some little holes, and most of these holes are happening over the genes, okay? So we see very different patterns. Not only that, now this CHH mark is not necessarily in the same places as this other mark. So we've learned a number of things from looking at Arabidopsis. They won't all translate. And I can show you, this is a recent paper from Bob Schmitz's lab. I want to make a couple points here. So number one, you can start to look at properties of the methylome and what types of bases are methylated across a lot of different species. They have 34 species. There are similarities among them, but there are also a number of differences. The other thing I'll point out is, so this is just correlating genome size to some of these properties. This one here is maize. So most of the genomes that have been looked at for methylomes are relatively small genomes. We're still getting that part of what happens in a larger genome. Okay, so in maize, one of the ways to think about methylation of the genome is to divide the genome up into 100 base pair regions and ask how much methylation of each type. If you look, what you'll see is that CG and CHG, over half of the genome, or sorry, the majority of the genome has over 50% methylation. Only a small subset of regions have very low, and really this is essentially a bimodal. You're either high in methylation or you're low. There's very few regions that have 20 to 40% methylation. Okay. CHH is a bit different. In most regions of the genome, it's very low, and there's only a small number that are somewhat higher. If we look at multiple contexts at a time, so we can classify the genome as having all three types of methylation, CG and CHG only. CG only being unmethylated or intermediate. If you look at the maize genome, the vast majority of this genome has these types of methylation without CHH. The unmethylated portion of the genome is relatively smaller. In Arabidopsis, you see a very different story. So the majority of the Arabidopsis genome is unmethylated. This orange region that was predominant in maize is now a very small portion of the genome. And how these are laid out on the chromosomes are quite different. So here I'm taking chromosome 1 from Arabidopsis, chromosome 1 from maize. In Arabidopsis, you see a very distinct type of chromatin around that pericentromeric region. In maize, there's a little bit of a bump when you get to the middle of the chromosome, but in general, you see all types of domains throughout the whole chromosome. Okay, so there's not as much segregation to a portion of a chromosome. Now, just as you think about this here, another way of thinking about this is that these global patterns are due to a composite of what happens at genes and what happens at transposons. So in general, genes have a valley. You drop down and have lower methylation of gene. Transposons go up, there's a peak of methylation. The three different colors of lines here are showing you CG, CHG, and CHH methylation. And the dashed lines are Arabidopsis, the solid lines are maize. So in both species, you see similar trends. They both have methylation that goes up. There are some differences, and we'll get into some of those in a second. One thing I want to emphasize, and I'm not going to tell you all of the details about this, but I'm somewhat lying to you here by giving you just two methylation profiles for transposons. If you break transposons into different subtypes, so class 1 or class 2, and then different superfamilies within that, you'll find that they don't all have the same patterns. So transposons, I shouldn't be lumping them all together and just calling them one thing. And perhaps the best way to show you is just look at these two families here. If we look at CHH methylation, this family clearly has strong levels of CHH methylation within those elements of the genome. This family here, which is another DNA transposon family, has virtually no enrichment of CHH methylation. Okay, so one of them attracts this type, one doesn't. I think we're still struggling to figure out what the features are that would predict which type of chromatin they have. But I want to just emphasize that not all transposons are being treated the same. Now, I want to pick up two interesting differences that we noticed between what the maize profile looked like and the Arabidopsis profile, because they illustrate, in some cases, they're helping us to learn what's going on and what some of the functions of methylation are, especially in larger genomes. So number one, if you look at CHG, the red line, you'll notice it's pretty much flat and virtually absent throughout the whole gene of an Arabidopsis gene. In maize, we actually see a bit of a bump. So there's CHG in the middle of maize genes. CHG methylation is often considered a fairly heterochromatic mark. So it was somewhat surprising to see it in the middle of a gene. The other thing that was a bit surprising was that CHH, the one in green, is considered a mark of de novo or RNA-directed DNA methylation. It has this bump right in front of and behind genes, so it's unclear why promoters would be targeted for this kind of methylation. So let's look into these two questions in the next slides. So the first question is actually pretty easy. It turns out that in maize, 10% of all maize genes have at least one transposon greater than 1,000 base pairs somewhere in that gene. Now, almost all the time it's in an intron, sometimes it's a UTR. When you get so here's a large intron of a gene, here's a transposon in that intron, it has very high levels of CHG methylation throughout, okay? So essentially the reason we're seeing that presence of CHG is because 10% of our genes have these methylated transposons. It turns out if I look at the methylation levels of those transposons that are in genes, they're just as highly methylated as transposons elsewhere in the genome, okay? What's somewhat surprising, or I guess was unexpected initially, was the fact that these don't seem to affect expression at all. Genes with these inserted in them, if anything, they tend to be higher expressed than other genes in the same genome. And if we look at allelic variants, so one allele has a transposon insertion that has heterochromatin, one does not, we see no consistent pattern for which of those two alleles would be more highly expressed than the other. So plants can tolerate the presence of these biochemical marks we would call heterochromatin. Transcription can elongate right through those without too severe a problem. Now, there are some very interesting studies in Arabidopsis from Tetsuji Kakatani that suggest that the ability to process transcripts through here is an active process, and there are genes required to do this normally. But normally, plants have that pathway functioning, and they can tolerate these things. So our definitions of heterochromatin, you couldn't get expression, don't really fit with this idea. Okay.
How do you determine that it's heterochromatin then?
Well, so that comes back to how you want to define something's being heterochromatin. If you want the classical cytological definition, just dark staining or light staining. If you want, so in practice, what most people are doing now is they use a particular chromatin mark. Some people like to use this H3K9 dimethyl mark. Some people like to use CHG, and they just say, whenever that mark is there, we call it, quote, heterochromatin. That doesn't necessarily mean, even though then heterochromatin is less likely to be expressed, it doesn't mean it can't be expressed. It's not an exclusive property. So I think part of the problem is we use this word heterochromatin for multiple types of things, and it doesn't entirely fit for all of them. Okay, so the other question was, what was going on with this CHH methylation in gene promoters? And I want to point out, this was first noted by Jonathan Gent and Kelly Daw. And when they first said this, I thought, that does not seem right. And they were very much correct. And my thoughts of what would happen were entirely wrong. So Ching Li and my group did a number of experiments to try to ask what was happening with this peak. And I'll point out in this one here, the CHH methylation is on a different scale than the other two contexts. But she wanted to know what was happening with this metaplot peak. Is this because a subset of genes have a bunch of CHH methylation, or all of them do? Was it a broad, a couple hundred bases of peak, or what was going on? Whoops, I pushed the wrong button. So one of the first things that she found was that about half of all maize genes have a region of high CHH methylation in their promoter. And the reason that we saw this so easily and that it hadn't been looked at in Arabidopsis is not because it doesn't exist, but because of the proportion of genes. This is a figure from that paper looking at 34 different plant species, and in Arabidopsis, only about 10% of genes have one of these elevated CHH patches in a gene promoter. In maize, it's slightly over 50%. okay? But if you look across all these species, most of them have some level. Maize is just on the high end, which made this easier to spot. So as we started to characterize these, one of the first things she looked at was those genes that have them. If we just plot each gene here is a line in this plot, and the more yellow it is, the higher the CHH methylation. And what she found was that there can be a region of high CHH anywhere from right near the transcriptional start site through a couple kb away. We, by definition, required one within 2 kb in this plot. But there tend to be very small regions, so it's not spreading over a large region. And what she did then was she took these little yellow boxes, and instead of making a plot that's centered on the start site for the gene, you center the plot on this region where that methylation goes up. And this is how it changes. So now we have a very strong peak. So before it was only up to about 10%, but now it's up to about 30%. So if you center on that region, you get a very high CHH. And this is exactly the place where CG and CHG are very high away from the gene and then drop as they get near the gene. Now, what controls where this happens? It seems that it's happening at the edge of the transposon that's closest to that gene. So the CHH methylation is happening over a transposon, but only the edge near the gene. This further away edge doesn't get that property. And interestingly, this tends to happen only for genes that are expressed, or it's more likely expressed genes than silenced genes. So while we normally think of methylation being associated with reduced expression, this methylation in the promoter is actually more likely if the gene is expressed. Now, not only does methylation change at this site, when we looked at other chromatin marks, so in gray you have chromatin accessibility. So if we line those genes up, we have more accessible chromatin right starting at that point towards the gene. H3K9 dimethyl, which is usually associated with silencing, drops right at that position. H3K4 tends to go up. So this position here that we're marking with CHH, that's how we defined it, is a place where transitions are happening pretty strongly. Now, there are a couple ways you could think about what this might be doing. So one of the ways to think about it is that having this CHH island, it's a place that it's helping to maintain the gene activity because it prevents the silencing and the chromatin marks that are in that transposon from spreading out into the gene. So maybe this boundary is there to make the transposon keep to itself. Alternatively, another hypothesis is that the boundary is there not to protect the gene, but to protect the silencing of the transposon from the activity of the gene. The way we've looked at this is using a couple mutants that Vicki Chandler's lab found. These are called MOP1 and MOP3, and they both affect components of the CHH methylation pathway. So in wild type, we had a CHH methylation patch here. It's entirely absent in these mutants. What we see is when it's absent, we actually lose CHG and CG methylation further back into the gene. This is compatible with the idea that the CHH island is there to keep the activity of the gene from being able to spread into the transposon. Okay? So if you have a genome with lots of transposons near genes, you're going to turn those genes on, it's going to be important that you make sure the transposons stay off. Okay? It seems that that's the function of putting this patch of methylation there, recruiting this machinery to that region. Okay, so that was my stories about where methylation is in the maize genome and what the patterns look like. For the next chunk of time, we're going to talk about where variation comes from, what impact variation has on this. So if you think about a particular region of the genome, and this region could be in an expressed state with very low levels of DNA methylation or highly methylated and silenced. There are lots of different sources of variation. So spontaneous changes could happen, stochastic changes in methylation. You could get genetic changes. These could be changes at that site or elsewhere in the genome. You could do wide crosses or have polyploids that would influence methylation. There are also a number of induced sources of variation. These could be mutants. So in Arabidopsis, we have lots of different mutants that can completely remove types or subsets of methylation. If you use those as crosses, many of those losses are stable and you can create a new heritable variant. You can do chemical treatments. These work with varying levels of effectiveness. Tissue culture actually is a treatment or environment, depending on how you want to think about it, that seems to have a higher rate of these shifts. And there's also potential for targeted editing using epigenome editing reagents. Now, I've left environment and development off of here, but we're going to talk about those in a second. So there are a lot of questions about how variable a methylome is and what are the things that trigger its variation. And I want to talk about a couple of these. So first of all, this idea that methylation is highly variable among tissues. Some of the initial idea was that methylation is associated with gene expression, and therefore if gene expression is changing during development, methylation must be changing too, and that's the main feature. I don't think that's true. So here, what I'm showing you is 4 different tissues of B73 and Missouri 17, and they're clustered by their methylation levels. What you notice is, regardless of what tissue you came from, B73 is much more similar to other B73 samples than to Missouri 17. And these two are pretty poor data, which is why they look different. Now, I'm not telling you that there aren't changes in the methylome during development. You can find a couple here. There are very clear examples in plant reproductive tissues, endosperm or pollen cells. There are nice examples in fruit ripening, where methylation changes can happen in development. However, compared to the level of variation between genotypes and other things, it doesn't seem like it's a major change that is occurring during development. At least it's not the primary driver of gene expression changes during development. There's also this question of what kind of variation we see following an environmental stress. So if we wanted DNA methylation to give us this potential Lamarckian acquired characteristic, you would be asking for plants to be able to have some environmental stress that would trigger a methylation change. And the way we did this experiment was we took young seedlings. We essentially brought them to a near-death experience. You can't see here, but this plant is very unhappy. And then we let the plant grow in normal conditions and take the last leaf of the plant. So the cells in that leaf, their progenitor should have been in the meristem when we did the treatment. So we wanted to ask, could we get mitotic, these heritable changes in methylation? We also had a similar experiment where we were taking tissue culture samples. What I can basically say is that we don't see, I took that out. So we don't see evidence that these ones were changing methylation. So I see differences among individual plants that either were subject to these stresses or controls, but the number of differences between 2 control plants is the same as the number of differences between a control and any one of the stressed plants. And there's nothing in common among plants that went through the same stress. Okay? So we don't see, at least that these stresses were triggering changes, reproducible changes in methylation or higher rates. Tissue culture is a little bit of an outlier. Here we have several hundred regions of the genome that do seem to be affected. Often they're consistently affected in multiple independent cell lines. So tissue culture clearly is doing something. I'm never sure how to think of tissue culture. Is it an environment? Is it a stress? Is it it's a pretty unusual treatment you would do to a plant. And there is very nice evidence from the idea that you could put plants in a tissue culture, you get phenotypes. So the idea that phenotypic variation happens from tissue culture has clearly been shown, and it's heritable. And I guess I'll mention palm oils now. So that's the reason I'm going to talk to people about palm oils, is because most palms go through tissue culture, and you get variants that are coming out. And at least some of those have been shown by the group in Malaysia and Rob Martinson's group to be epigenetic in origin. Okay.
Before you do
yes.
that slide, so when you did this, you did methyl dip.
Yes.
Do you think if you went back and looked at it at nucleotide resolution that you might see something different?
So I don't think I would see anything different for CG and CHG methylation. What we've found is that we can pretty well reproduce many of the patterns for those types. CHH methylation, I struggle with in various ways. It's at a lower abundance than the other types. So if you remember CHH methylation, most of the genome didn't have it, but when it's there, it's at like 20 to 40%. Other studies, so Ryan Lister's group and others, have found environmental changes that were triggered in methylation. But when they looked really carefully, what they found was that they happened to be at genes that got turned on by that stress, and often the methylation change happened after the gene got turned on. So in other words, the gene would turn on and it would allow methylation to now be targeted to a region. When the gene turned off again, those weren't heritable changes, because if the gene returned to its normal level, the methylation went back again. So I don't know that we would find very many heritable changes. And I'll come back to this later, but this idea that you would have substantial variation following environmental stress would suggest that methylation wouldn't be all that heritable. Because if you could pass through different environments, you should end up at pretty different points before too long. And really, most methylation is quite heritable, even across generations. So if the environment were really so easily messing with things, and I want to back up and say, this is methylation I'm talking about, DNA methylation. If we look at some of the histone marks, I think some of those are probably changing and probably are different in these plants even at maturity than other things. But I think plants are reserving DNA methylation for a more stable mark. Okay, so in terms of sources, those we're looking at environmental, developmental sources of variation. I want to talk about some of the genetic sources now. Since I started graduate school, which was a fairly long time ago, I've been trying to do this project, which is to essentially collect mutants that would affect methylation, so I could tell you what the role of DNA methylation was in plants, or particularly in maize. So I've done a lot of reverse genetics, finding different genes, collecting mutant alleles. We've attempted RNAi. Other groups like Vicki Chandler, Karen McGinnis, Jay Hollick, and others have been doing forward genetics to look for things that affected epigenetic silencing. And they have found things like the MOP mutants and others. So we've collected all these mutants. We have done bisulfite sequencing on a lot of them, and these two dashed bars here showing you the range of methylation levels we see in wild-type plants of different natural variant lines. And what you'll find is none of our mutants do much here. A couple of them knock down CHG methylation a little bit, and a couple of them knock down CHH. But these are not things to be particularly excited about, right? If this were Arabidopsis, Eric's mutants that he found years ago would be down near zero here, some of these mutants, okay? So we have not been able to really perturb the methylome substantially. And if I take two paralogs and I attempt to create a double mutant, I cannot recover that double mutant. So maize, there's now evidence in rice from several groups. Tomato has similar evidence that if you really affect methylation in most plant genomes, you struggle to get back viable plants. So Arabidopsis might be a bit of an outlier in how easy it has been to recover really strong loss of function mutations that affect methylation. This is a bit of a problem in terms of getting conclusive mutants, and why, for the rest of the story, I'm going to tell you about natural variation instead. Okay? So when we have natural variation in methylation, I'm showing you two lines now. B73 are the top three tracks, and W22 are the bottom three. And all these regions in yellow are nice places where the methylation level of the two lines varies substantially. So this is looking at about 4,000 regions in 90 samples here. And this helps me to kind of re-illustrate some of the things I just mentioned to you. So here's a block of B73 plants. And in this block, I have a number of different developmental tissues. But what you'll notice is this block looks pretty darn similar to each other. So even though they're from very different developmental tissues, that wasn't separating them into different clusters. Over here, this is a block that's gone through tissue culture. There are some differences among them, but they are different from other samples. This block here is a whole bunch of teosinte accessions. They're not identical to each other, and they're not genetically identical either. They're from accessions where they're still, they're outcrossing. So we have lots of variation, but by and large, this tree can be reconstructed by genetic relationships as much as anything. So we don't see massive changes among tissues. And this is with bisulfite data, so we should be able to see some of those. Although this is only CG methylation, so it's not that CHH. All right, so natural variation. I'm going to tell you about how much there is, what we think about causes for it, how it's inherited, and what the effects are. First thing I should mention is we only get to look at about 30% of the genome. So when we compare two different lines, even if I'm only looking at B73, I get to look at about 55% of the genome because of repetitiveness. The highly repetitive transposons I can't look at very well. If I compare two other inbreds, the polymorphism rate on top of that difference in the uniqueness constraint gets me down to anywhere from about 30% to some genotypes, it's 40% of regions of the genome I can compare. When I look at DMRs, I find tens of thousands of CG or CHG and thousands of CHH DMRs. Those are big numbers. That makes you happy, like, oh, things are changing all over the place. But if we look at this as a percent of the genome that we can test, for CHH methylation, it's really somewhere between about 0.08 and 0.12% of the genome that is changing. The majority, about 99.9% of the genome, isn't changing for this mark in any two of these lines. And even for these other types, we're only looking at about 0.5 to 0.8% of the genome. So greater than 99% is stable and hasn't changed. We now have looked at a larger panel, so about 16 lines. In total, when you sum up all of the differences, it's only about 5 to 7%, depending on the context of the genome, that has varied. 95% has stayed the same. So all of these DMRs we have, how are they inherited? How stable are they? We've looked at it a couple ways. So one way we looked at it was to take those DMRs and use single nucleotide polymorphisms nearby to do a local association scan. So this is not GWAS. This is just saying if I take a region and I only look at nearby SNPs, can I have a nearby SNP in LD with this chromatin property? And the answer is almost exactly 50% of the time, yes, there was a SNP that was linked. The other half of the time there wasn't. Now, having a SNP that's linked doesn't say that SNP does it. It could be that that SNP is linked to a transposon or something else. Or it could just be that this is a very stable change that's been there for a long time and happens to be an LD with a nearby SNP. We also looked at biparental populations and looked at inheritance of methylation there. What I would say is most of the time, the genotype of a region of the genome will tell you whether it's methylated or not based on what was in the parents. So in other words, if you inherited the allele that had been methylated before, it stayed methylated. Very rarely did we see a disconnect between what we expect based on the local genotype and what we actually observe. So these are words that Eric Richards, or concepts that he came up with a long time ago that have been exceptionally useful for how I've thought about epialleles and changes. So the idea here is that if you have a region of the genome that is expressed and a chromatin change happens that changes expression, and this happens in the absence of any genetic change, that would be a pure epiallele. So it's purely epigenetic. In contrast, if there is some sort of genetic change, so a transposon insertion or a structural rearrangement, this could then trigger a change in the chromatin of the low copy shared sequence that could either happen all the time when that sequence changed, or it could lead to a poised state. In this case, you can survey chromatin and find a difference, but there's also probably a genetic contribution to what's happening here. So it's not purely epigenetic. So we took all of those DMRs and we started asking, what's the evidence that this could happen versus what's the evidence this might be happening? We can find evidence for both, so it's not entirely satisfying, but we'll start with this one first. So the way we've looked for these types of things is by taking the fact that maize is an outcrosser, but the lines we're looking at have some portions of their genome that are identical by descent. So if we look at regions of the genome that are at least a megabase of size that essentially have no SNPs, we find that the rate of DMRs, the percent of the genome that's DMRs, is shown in red. So for those regions, it's about 30% to 50% the rate of other portions of the genome. So there's reduced rates of DMRs when the genome sequence is similar in that region, but it's not zero. So we think that they're there, but they're not as common as they are elsewhere in the genome. When we take the other tact and we say, let's test this idea. Let's ask if transposons are inserted in one line and not another, can we get a methylation change nearby? I'm going to summarize it just with one picture. Basically, in this picture, the plus or minus is telling you whether a particular transposon is inserted right here. The haplotypes with that insertion have high levels of methylation nearby. The haplotypes without don't. So we would call this a DMR. And if we didn't know about this transposon insertion, we wouldn't know that it was being caused by something else. But I think this is due to a transposon. Now, I don't want you to leave thinking all transposons will do this. It turned out when we looked per family, only about a third of all of the transposon families had any influence on the chromatin nearby. The other 2/3 of them don't, and we still don't know what makes those 2/3 different from the 1/3. So they're different types of elements, they're different types of families, but it seems like some transposons influence neighboring regions, others don't. Now, do these DMRs matter? What kinds of effects do they have? I'm going to show you two different ways of looking at this. So the first way we had of looking at this was we took all of those DMRs and we found 2,375 genes that were located near a DMR. We had expression data on the same tissue. And so we said, how often when there's a change in methylation is there also a change in expression? In about 10% of those cases, we have a significant negative correlation. There are another 53 with a positive correlation. So that is that if it's negative, the methylated allele is lower expression. Okay? If it's positive, the methylated allele is higher expressed. I don't quite know what to do with that number. So it's certainly true that not all of the DMRs are affecting expression, but it's also 277 genes is a decent number of genes to be on or off, and that's some level of natural variation there. In total, this is a little under 1% of all genes in the maize genome, but 1% of genes being influenced by epigenetic variation in a population, in this case of about 50 lines, that's a substantial number of alleles that could be influencing traits. The other way we looked at this, which I think I like better after I've thought about them both for a while, was to start not from the DMRs, but from expression change. So if I take all of the genes and I sort them into being non-differentially expressed, being slightly changed, 2 to 5-fold change, all the way to being on-off states. And then we look at how often do they have a methylation change over their promoter. What we find is, this is for CG and CHG methylation, that only the genes that are on-off or greater than 10-fold change have a significantly higher rate of methylation changes compared to non-differentially expressed genes. So in other words, if methylation is changing, the type of effect it's going to have are very severe changes in expression. So this subset that are being affected, and in this case, if you look at genes with a greater than 10-fold or an on-off difference, it can be as many as 20% of those genes that are associated with the methylation change nearby. Okay? So really, I don't think methylation is doing a lot of the subtle quantitative changes. It's doing more strong changes in expression. Now, this is looking at genes that are varying naturally for methylation and varying for expression. The other interesting idea about methylation is that there are things that are invariable, they're silenced, and that we would refer to these as cryptic information. And different groups, so this is from Bob Schmitz's group, they were trying to count genes that they thought might have been putative epialleles, and they were counting them in a number of species. So they estimated there might be about 5,500 in maize. We did this in a different way. When we did this, we were using expression data from a lot of different individuals, as well as some methylation data. And I would estimate that there were about 4,000 potential epialleles. Many of these, so about 1,500 of those, already exist in both states in nature, as near as we can tell. So they're expressed in some lines, they are not expressed in others. The rest of them, though, about 2,000 of these, in the 50 lines we have looked at, they are methylated and silenced in all of them. And so there's potential information there that if you could release that methylation, you might get some change in phenotype. Okay, I realize I'm about out of time. So this is the summary of the methylome part. So I think that there's a lot of potential for using this to guide functional analysis. We don't see a lot of variation in development and response environment. This is not saying there's zero, but it's saying we're not seeing massive changes. And we see a lot of natural variation, and we think this can contribute to variation for expression epialleles. Now, if you will give me like 3 or 4 more minutes, I'll go quickly through another story. Not this one, I'm gonna skip this one. So I'm gonna talk about TEs and expression just for a little bit, because I think this is cool and it ties into this other stuff. So Sarah Anderson is a postdoc in my lab, and she's been developing pipelines to allow us to quantify transposon expression. Often we don't do this because many of the reads we care about can actually map multiply. They map to multiple families. So what she finds is if you look across development and quantify how much of the genome can be, or how much of the transcriptome is coming from TEs, it's anywhere from about 3 to 5% up to almost 20%. So a decent amount of transcriptome. Now it's not the 65% of the transcriptome, which is how much of the genome these things are accounting for, but it's not 0. So often people think of transposons as all being silent. They're expressed. They're often expressed in dynamic patterns. So there are a bunch of families, all of these ones up here, that are only expressed in pollen. They're not expressed elsewhere. There's a bunch of families, the second group, that are expressed pretty much everywhere but pollen. There are also groups that are expressed in other particular tissues. So these aren't just noise, they have particular patterns of expression. Those patterns can include stress responses. So we have cases where particular stresses will cause an element to turn on, or multiple stresses. Why do we care about this? So often we think about transposons as disrupting things. They land somewhere, they break a gene, they break a regulator. But transposons could also bring with them new information. So this idea that they're helping to shape transcriptional responses by where they insert. This is a concept from McClintock, suggested many years ago that transposons would act as controlling elements. There are nice studies in Arabidopsis that suggest this. There's some concepts from tobacco and rice as well. In all of those cases, though, you have relatively few numbers of transposons in the genome, and not a lot of them are near genes. So we looked at this in Maize. Irina Makarevich did this, and what we find is basically there's about 20 families of transposons that are significantly associated with whether or not genes nearby are upregulated in response to particular stresses. So the idea is that as this transposon moves, it can create a new regulatory response for a gene. With the help of Michelle Stitzer and Jeff Rossabara, what we could show is that if you look at polymorphisms, so the pluses and minuses tell you whether or not the transposon is there. In alleles with the transposon insertion, you get upregulated expression in response to that stress. The alleles without it are not responding. And here's looking at about 25 lines. Half of them that don't have, that have the insertion, half that don't have it. So only when you have this insertion do you gain this response. So it's hard to think of how, from a single base change, you would create these new responses. Transposons give you this option that as they carry their own enhancers and regulatory information with them, as they land in the genome, they could affect nearby genes. So, sorry I went through that one quick, man, but I'm trying to get done about on time. So we can look at TE expression. There's dynamic expression of TEs themselves, and I think they're influencing nearby genes. And I think I've mentioned people as we've gone. Appreciate funding and all of the wonderful people I work with. So take any questions. Yeah.
So in the sort of last story, when transposons affect, like, increase the response of genes in response to expression of genes in response to a stress. It's a two-part question. Are they typically beneficial to that stress, like for the plant? You know, are they stress response genes? And two, does that indicate that part of what methylation could be doing is actually providing information for where they should insert, where transposons can and cannot and should and should not insert, not only the expression, but the reinsertion?
So let me start with the second one. So first of all, I don't think the methylation is changing with that stress when that happens. Now, your idea that maybe transposons are more likely to insert into regions with a particular chromatin, I think that certainly is true. You see some transposons that really prefer more open sites for insertion. Others seem to prefer to land in heterochromatin sites, depending on how you quantify those. For the first part of the question, So there's multiple answers. So number one, no, I don't think that they're doing beneficial things. I think that in most cases, if you turn on a gene in response to, say, cold stress, it probably doesn't hurt you, right? There are probably not a lot of genes that turning them on are detrimental in that case. So I don't think there's a big cost per se. So if this is simply a expression mutagenesis that's happening naturally, every now and then you would have an insertion that would land, that would give you something beneficial to the plant, and that would then be selected for over time. Over long periods of time, you'd get to the point where you can't even see the transposon. The transposon would be removed by mutation and other things, but the enhancer that it brought with it would have been maintained by natural selection, right? There's a little bit of a problem with this, and that is that if we do look at Go enrichment terms, there's some enrichment for stress-related genes around this, but often the stress-related genes were defined by expression patterns. So it's circular logic. The fact that they come on in stress defines who they are, and the fact that this lands near them and is causing them to come on in response to stress isn't very helpful. So I don't think that the genes being responsive to this are necessarily more beneficial.
I'm wondering, related to that, about the methylation changes in response to stress. They didn't find a lot of changes. For your stress, it looked like it was a pretty severe stress. So I'm guessing it was a short duration. I'm wondering if there's any reason to think or to hypothesize that maybe methylation changes maybe occur more for long-term stresses, which would be more beneficial from an evolutionary perspective.
So we basically did the stresses, I think they were 8 hours once a day for 7 days. So we did try to give them repeated levels of stress, but it wasn't that long term. I mean, the problem is you have a finite life cycle, and so if you want to, we had to think about that. The other thing is, I've struggled from the standpoint of like, why would a plant want to remember? So if it's hotter this year, does that mean it will be hotter next summer? Not necessarily. The weather changes. Is it, so I'm trying to think sometimes about what stresses you want to remember, what stresses really it would be important to hardwire versus maintaining a response to those stress. So I'm not sure that these stresses are ones that would be reproducible year after year.
It's probably, I guess, very different too between like something that lives for 1 year versus many years, right? Because especially if you live many years, not stress for like a few days or a few months, yeah, you probably don't remember that. But if you're experiencing a stress long term, like maybe for the entire lifetime of a plant, or for many years.
And so that's where perennial species might have very different patterns. So a branch of a tree that is shaded all the time will probably be shaded next year too. So in cases like that, being able to remember mitotically in that individual might be different than to the next generation.
You mentioned during formation of epialleles, that section, that wide crosses and polyploidy you mentioned. So when you say polyploidy there, are you talking wide crosses allopolyploidy, or are you talking the actual process of genome duplication?
Yeah, so that's something we haven't looked at in maize, but there have been a number of studies, particularly in wheat and other things, looking at some of the original allopolyploids. The autopolyploids, there's some publications out there that have found some changes following autopolyploidy. There aren't as many that I'm aware of, and there aren't as extensive of changes as people have suggested in some of those allopolyploid events. The overall idea that in one sense, that wide crosses and polyploidy is kind of a subset of the genetic changes part. So in one sense, if there is a small RNA producing locus that's in one haplotype or one parent and not the other, in the heterozygote, it would have the potential to influence the state on both alleles. So the idea is that you're gaining new signals, and often it's new genetic information through that process. It's not to say that there isn't enough stress or shock from the process of formation that could cause it as well. When we looked in maize, the closest we looked at was doubled haploids. And there we didn't see anything changing in a doubled haploid. So going through that, you could think of that as a stress being a haploid file and doubling. We didn't see that leading to changes.
Yeah. So I'm wondering, so do you have any idea about what the regulating mechanism about the differentially correct transposon for regulating the stress response gene? So, I mean, is there any indication or something like that, whether there any physical interaction or there any localization decrease or something like that?
So we looked at different families. Sometimes the transposon needs to be pretty close to the gene, a couple kb. That there was certainly a limitation how far away they could be. Mechanistically, we haven't looked at what's happening, but what we think is happening is you have an enhancer sequence. In almost every one of those cases, the transposon itself is also upregulated by that stress. But we think that the enhancer can act not only on the promoter of the transposon, but the enhancer that's contained within the transposon could also act on the promoter of the gene. So we think it's acting as a local enhancer. We don't see any evidence that transcription is starting in the transposon and reading through to the gene. And it doesn't matter which orientation the transposon is in relative to the gene. So that's why we think it's acting as a local enhancer.
Okay. No further question, then thank you again.
This has been a production of Cornell University, on the web at cornell.edu.